Good morning, everybody. Welcome. I'm Tom Heeman. I'm joined with my teammates here, Abraham Vazirabad, Nicholas Tewalt, and Jacob Meinking, and we are the Adam family, and we are advised by Doctors Hasper and Ashram. Our project is engineering Atom-17 constructs towards variable functionality of TACE. So TACE and Atom-17 are kind of used interchangeably, so we're going to stick with TACE from here on out. We used Atom-17, which is the gene name, just for the inspiration of our team name. So first, our outline. I'll cover the project overview, kind of what it is, why it matters, just sort of the aims and objectives. Then Abraham will take the project progress and some of the results that we had. Nicholas will pick up for procedures and project continuation. Jacob will take scale up and sort of the industrial and financial analysis, and then questions maybe at the end. So first off, what is TACE? TACE is a protein, and it's a shed-ACE class protein. So the suffix ACE just means it's an enzyme that differentiates it from signaling proteins and structural proteins, so it's actually doing something. And then the prefix shed is simply the verb, meaning to tear with force. So it's not, so it's actually like doing the work. It's imparting work on the system. So what it specifically does is it cleaves ectodomains of proteins. As you can see, it's a transmembrane protein, and all these transmembrane proteins have these transmembrane domains that anchor them to the surface of the cell. So we know by the lipid or the fluid mosaic model that all these can migrate about the surface of the cell. So what happens is these growth factors, this is a growth factor here, it's a little difficult to see, but it's in an anchored position. So when it's anchored, it's not actually in the system. It's sort of like a resting state. So it's produced, it resides on the surface of the cell, but it's not actually, the signal isn't active. It can't be received by anything. So TACE cleaves it right at the surface of the cell, thereby liberating it into the extracellular space. So at this point, it can interact with signal receivers of the same cell or other cells. So in this case, it comes back down, it's received by the receptor protein here. The signal's transduced back through the cell, and in this case, it's a growth factor. So it could be promoting cell growth or epithelial sheet migration, which is of particular importance, and we're going to touch on that a little bit later. One thing that's important to note is that TACE is actually an acronym, and it stands for tumor necrosis factor alpha converting enzyme. You can barely see it right here, but it stands for TACE. So what is tumor necrosis factor alpha? Well, it's probably the most important and exciting growth factor or signaling molecule that's cleaved by TACE. So this is probably because of its, this is because of its involvement in so many excellent targets, medically speaking. So first off, inflammation it's very important in the innate immune response, and that's before your immune system is recruited. As well as it initiates apoptosis, and that represents a great target for inhibiting tumorigenesis or as a cancer treatment. In excess, it's involved with arthritis, and the deregulation is involved in arthritis or Alzheimer's disease and cancers. So this represents a great target for medical therapies and things of that nature. So first off, discovering the cause. This is when we sort of discovered the cause of our project. So it all starts with this little guy, the common mouse, Mus musculus. There's a known point mutation that occurs in the wild type genome, and it's an ATG or an ACG to an ATG. And in the entire gene, everything is preserved. It's just one amino acid, or just one nucleic acid point mutation that results in amino acid substitution. So you go from the wild type, which is the normal mouse that we all know, to this, what we call the Woe phenotype, that's waved with open eyes. So the fur is actually waved because of the epithelial sheet migration is affected. There's reduced protein activity here. So the fur is waved, and they're born with open eyes because the sheet migration is supposed to, during embryonic development, draw the eyelids closed. but this doesn't happen, so they're born with open eyes. So they also have a couple minor defects. There's a couple eye defects. Obviously, the fur, if you can count that as a defect, and there's a couple other minor fur defects as well. But they're still viable young. They still grow, they still eat, they can still reproduce. In the wild, they might not, but in the lab, they do. So this is a known point mutation that occurs. So if we look at our protein here for a second, it's a little difficult to see on this, since it's so whited out. But this yellow band here is the amino acid 265. That's where this mutation occurs. And you can see it's in just a free loop here, which might be, that'll be important in a second here. There's a large red section here that you can barely see. It's dark red. But what happens is in the experimentation, in our research that we found of previous research done, when this section here, if you can see, it's kind of like an orange section. It's supposed to be orange. When that entire part is excised from the protein, it's a missense protein, so it doesn't have any taste functionality whatsoever. When we look at that, what we have is down here, that excision is embryonically lethal. At that point, the young do not survive birth. So that's when there's absolutely no taste functionality whatsoever. That low phenotype is very, very low in comparison to the wild type. If we were to assign numbers, it's something like 0, 5, obviously wild type is like 100% functionality, and then overexpression of taste is bad. If there's too much functionality, that's linked to some cancers. So this is kind of the gradient that we think is happening here, and this is our project hypothesis. Between the wild type and this low phenotype, this is our design space, and this is where we're aiming to design our two constructs to fall as far as like protein functionality is speaking. So this is sort of the overview and what we're aiming to achieve here. So to decide on our targets, our design targets, we need to look at residue 265 and what's happening here. So obviously the wild type is threonine, that's at full functionality, and this low, very, very low functionality is methionine. And again, here's our protein. So we need to look at these and compare them and see what characteristics they share and what they do not share, what makes them different. Well, they're actually much more different than they are the same. So threonine obviously is lacking many characteristics that methionine has. The fourth factor that we could have put in here is ionic charge, but neither of them have that. We could have tested that, but it would have been rather arbitrary. So what we aim to do here is emulate some of the characteristics of methionine here. So our first target was valine. And what this does is it shares the hydrophobicity, but it is also very small and it does not have any sulfur. Then our second target is cysteine, as that shares sulfur and again is very small and it is not hydrophobic. So in each instance we're testing one variable because we don't know what is primarily responsible for this link between functionality and this base here. And then by proxy we sort of test by size. If both of these come out and they are exactly like the wild type or very similar to wild type, then we can reasonably assume that it's just the sheer size of methionine that is responsible for this change in functionality, and we could throw other large amino acids in there and expect the same thing. So this is our overall hypothesis here, that our two targets that we are designing, the valine and the cysteine construct, will fall somewhere on this scale. This is sort of arbitrarily spaced. We don't know where these are going to fall. We do not know this relationship, so we can't really even guess as to where they will fall on this gradient. But that is the overall goal of the project. So our project goals are to identify the relationship of this residue 265 and its link to the protein functionality. This will most likely depend on its structural and chemical properties. Okay. So we also aim to produce our constructs. The valine and the cysteine are our two variable constructs, and methionine is our low construct, and that has to be made by us because we cannot harvest and do primary cell culture, so that must be made as well. We aim to quantify our findings and hopefully provide some kind of foundation for a legacy project. So again, the relevance. There's many applications in humans. The most exciting are the cancer, Alzheimer's disease, and the potential arthritis therapies. And the main focus is potentially gene therapies is what our overall project product would be. And we're focusing on tunable protein activity, and I want to stress that because we're not affecting the protein expression, because those expression systems are ridiculously complicated, and I mean, as we know from synthetic biology, it is very, very complicated. So all we're doing is we're changing the actuator. We're changing the protein expression itself. So the overall problem statement is that two constructs currently exist in nature. That's the wild type and the woe. And we believe that further engineering could result in a tool usable for the development of pharmaceuticals or gene therapies. So with that, I'll hand it off to Abraham, who will tell you how we're going to do this.
All right, thank you very much, Tom. All right, this is our team's process flowchart. It's an overview of all process steps, and our team will be discussing each step in detail. So here we have a condensed version of our team's subsystem diagram, and portions of this diagram will be used throughout our team's presentation. So first, let's talk about our team's preliminary studies. We modeled our region of interest, and our target is the amino acid position 265 in the TACE protein. We chose our 3 amino acid targets, and they are cysteine, methionine, and valine. Thirdly, we generated our list of materials. For example, we chose an immortalized keratinocyte cell line called RT101 for the mammalian cell culture portion of our project. Regarding our consumables, such as tips and flasks, we will use what we need from the Washington School of Engineering. And then finally, we generated a list of reagents and kits that we'll be using in this project. We then went on to obtain our wild-type TACE construct. It has been engineered to contain TACE, which you can see here in the yellow, and there are 2 reporter genes, which will be very important in downstream steps of the project. We then moved on to design our PCR and sequencing primers. However, once we began to design these primers, we ran into a constraint in that the existing sequencing, the existing sequence data was found to conflict. And this conflict was between the Invitrogen data, which they generated the construct backbone, and AdGene, who we purchased the wild-type TACE construct from. So here we have an alignment of the Invitrogen sequencing data and the AdGene sequencing data. And we see that the alignment near the TACE start codon, the alignment is identical, it's entirely the same. However, around 10 base pairs upstream of the start codon, the alignment becomes entirely divergent. So we found we needed more information about the construct sequence in order to continue with our design. And the solution to this constraint was to design reconnaissance primers to explore both the upstream and downstream regions of the TACE DNA. And the result of this exploration was that the Invitrogen sequencing data was found to be correct, so we used that. And with this information, we were able to design PCR primers. You really can't see them very well, it's kind of whited out. But each set, there's 1 set here, 5 sets in all, and they each amplify around 500 base pairs of the TACE DNA. And when we apply these primers to TACE DNA, we see they overlap one another. And this is necessary and relevant for the downstream step of sequencing. And if we zoom in on a specific portion of the TACE DNA, we see there's a 3 base pair region which is colored in yellow. And this is the target of our rational modification. And therefore, in order to rationally modify and alter the TACE DNA, we need to design site-directed immunogenesis primers. So our target amino acid is at position 265, as you see right here. And these primers are able to alter the wild-type amino acid, which is threonine, to encode instead for cysteine, methionine, and valine. After we design these primers, we then use them in the site-directed immunogenesis protocol. This method is used to redesign DNA in a very controlled and specific manner. This is a very important step, since this will generate our 3 engineered constructs of cysteine, methionine, and valine. And the image to the left you see here is an overview of the protocol itself. So we performed the mutagenesis protocol, and we acquired our 3 engineered constructs. And here's a table of the mass amounts we collected. As you can see, we have sufficient construct for all of our downstream steps. And the next section of our project will involve verification of the construct that we acquired. So we moved on to the quality control and validation portion of our project. Therefore, it was necessary to sequence the wild type and the 3 engineered constructs that we generated. However, the Milwaukee School of Engineering does not have the ability to sequence DNA in-house. So we therefore forged a relationship with the Milwaukee Health Department to sequence both our wild type and our 3 engineered constructs. So here we have a visualization of the wild type TACE sequencing data. We see that there are some white lines that are interspersed, and these represent discrepancy between the wild type TACE sequence and the sequencing data that we got back from the Milwaukee Health Department. It's quite possible that some errors accumulated via the polymerase chain reaction and the sequencing reaction that we performed. However, to resequence the wild type TACE DNA would have been economically constraining for us, and therefore the team chose not to resequence. So here we have a wild type TACE chromatogram, and a chromatogram is the end result of the sequencing process. We're looking specifically at position 265, right over here. And we see that at position 265, the bases that were called were an A, C, and G. And this encodes for the amino acid threonine. Therefore, this suggests that the construct that was sequenced is indeed the naturally occurring wild type, which is what we expected. So we then moved on to the validation and verification of our 3 engineered constructs. And we're looking specifically, again, at position 265, and we're studying the cysteine, methionine, and valine constructs. At position 265, we expect a TCG for the cysteine construct, ATG for the methionine construct, and a GTG for the valine construct. So let's look at each of these in turn. So we look at position 265 for the cysteine construct, and there's a GTG there. And this appears to be like the valine construct. For the methionine construct, there's an ATG there, which is what we expected. However, there are 2 missing base pairs around position 261. And finally, valine has the expected GTG. So let's look at each of these in more detail via their chromatograms. So we're looking at the cysteine construct. At position 265, there's a GTG. And therefore, the sequencer called this construct as a valine construct. So therefore, we need to revalidate the cysteine construct. A methionine construct, looking at position 265 again. The ATG is what we expected. However, there are 2 missing base pairs around position 261. What should be there are 2 sets of ATs in a row, an AT and an AT. That should be there. But there's only 1 set of ATs, only 1 set of ATs. And therefore, this leads to a frame shift mutation, and therefore, the methionine construct also needs to be revalidated. And finally, looking at the valine construct at position 265, there is a GT and G, which is what we expected, and therefore the valine construct is liberated for our downstream steps. And to talk about these downstream steps, I'm going to hand it off to Nick.
Thank you, Abraham. All right, so now that we have verified our constructs, as well as have a successful culturing of a keratinocyte cell line, time permitting, we would have pushed forward with a technique known as transfection. This is actually the physical uptake of DNA or our constructs into a cell line. Our team had identified 2 separate techniques. One is electroporation, and the other is liposuction. We'll go a little bit more into detail with these here. Let's start with electroporation. This actually uses microelectric pulses to actually perforate the cell membrane. In doing this, it allows the DNA to slip through the cell membrane and be incorporated into the cell line. As you can see here, the cell is fully intact, but once we apply this electric field, it allows these small pores to open up on the cell surface. Once this happens, you can't really see, but these little green squigglies, which are our constructs, actually slip through the cell membrane and are within the cell line. Once inside, we remove the electric field, and the cell then closes up again with the DNA remaining inside. With the DNA inside, the cell is able to express and utilize this protein. This technique is fairly effective and actually relatively inexpensive, which is good for our economic needs. The second one that we identified was that of liposuction. It runs on the same concept as electroporation. However, it uses a thing called a liposome, which actually encapsulates the DNA and through endocytosis is then incorporated into the cell. This technique is a little bit more effective than the electroporation technique. However, on the downside, it is a little bit more expensive. However, in the end, we did intend to use both techniques. Now that the DNA is within the cell and actually expressing, we want to be able to quantify the overall activity of our protein within the cellular environment. Our team had identified a kit known as an anaspect sensor-like kit. This runs on a fluorometric system, or the emission of light. Within the kit, it contains a peptide, which is TACE specific, which can be cleaved only by TACE. Once cleaved by TACE, it emits this fluorescence, which is then read on the plate reader, which we do have in-house. The amount of fluorescence given off is directly analogous to the overall activity of our constructs. Another technique that we identified was that of a scratch assay. It is more of a physical observation of how well our constructs are working within the cellular environment. As you can see here, once we plate the cells, we actually use a micropipette tip to scratch the surface of that plate, allowing for this open region to appear. Over a period of time, we would analyze and observe how well those cells actually move into that open region. As Tom had stated before, TACE is directly related to cell migration or sheet migration. So the more cells that move into this area, the more effective our constructs are. Once we have completely obtained these results, this would have concluded our project in full. Throughout our project, we encountered a few global constraints, which are listed here. In the initial stages of developing our project, we had hoped to use a variety of kits, but quickly came to realize that these were a little bit more expensive than anticipated. So in light of this situation, we had erred on the side of only 3 constructs instead of doing more, which would have required the purchasing of more materials and kits. We also researched and obtained kits at a lesser cost that were able to provide us results similar to those of the more expensive kits. We had also hoped and planned to use a human cell line in the initial stages of development, but due to MSOE regulations, we weren't allowed to do this. Therefore, we opted for the mouse cell line, which gave us a similar cellular environment to that of the human cell line. Also, a number of our kits that we use contain volatile materials, as well as our close interaction with potentially dangerous biological material, which provided us with a potentially unsafe work environment. To overcome these safety issues, we just review the protocols constantly, both before and during the experiment itself, as well as adhere to good manufacturing practices, or GMPs, that are related to proper disposal and proper use of these materials. And lastly, a portion of society does not fully agree with genetic engineering or the use of a animal cell line or animal testing. So a solution to this would just be thorough clinical trials, proven safety in both the short term as well as the long term, and catering to animal rights and regulations. So even though we encountered a few bumps along the way, we were able to overcome these without hindering any of our project's results. And so from here, I'd like to pass it off to Jacob, who will give us a little idea about the scale-up of this project.
Okay, so I'm going to take you through an overview of what gene therapy is and its market value, current good manufacturing practices and validation, some environmental and safety issues, and then I'll give you a description of our process from the primary recovery to final purification, and then give you a final cost estimate. So our process would be developing bulk TACE plasmid. So plasmids are covalently closed double-stranded DNA molecules, which can be used for gene therapy. Gene therapy is when you modify the genetic expression for therapeutic purposes. Both viral and non-viral vectors have been developed to efficiently deliver nucleic acids to target cells. However, viral methods have encountered safety and regulatory issues after half dozen deaths in early clinical trials. So plasmid offers a valuable gene transfer system in a market that is expected to have substantial growth. On the right here, you can see a pie chart of the vectors used in gene therapy clinical trials in 2012. Right here, it's supposed to be red, but of course we're having trouble seeing that, is the plasmid DNA, which makes up 18% of the clinical trials, coming in just under the adenovirus and retrovirus methods. So we would have a sizable portion of the market. So plasmid DNA, which is to be administered to humans and animals, must be manufactured under current good manufacturing requirements. So these are otherwise known as GMPs and are set in place by the Federal Drug Administration. And GMPs cover all aspects of production, from choosing raw materials to the final delivery of your product to the clinic. So if your product is not in conformity, it cannot be legally approved be approved by the FDA. So to do this, we develop validation, which ensures that the GMPs are met. So this gives you a reasonable, it gives you the purity, the potency, identity, efficacy, and safety that you need out of your product. It reduces the variability. Also, it reduces the number of failed batches, which decreases manufacturing costs. There are some environmental and safety issues that come along with common components of biological waste streams. These are buffers, acids, alkalis, and E. coli-derived organic chemicals, such as the cell debris and the media components that we use. However, these pose limited risks to the environment once inactivated. So for our process, optimization meant the minimization of waste so that we could minimize costs. So this is a process overview of what we designed using SuperCode Designer, which is a process design software. So we began with primary recovery, from fermentation to dead infiltration. Intermediate recovery, which included 2 precipitation steps to obtain the plasmid DNA. And then final purification using chromatography. So I'm going to go a little more in depth about each step. So we begin using fermentation. This multiplies the amount of cells we have, thereby increasing the amount of plasmid DNA. Then we harvest the cells from the fermenter, along with other impurities, and we centrifuge that down. This results in a significant reduction in volume, along with the removal of the most plentiful impurities, such as extracellular proteins, some genomic DNA, and lipids as well. So now we need to be able to obtain the plasmid DNA from inside our cells, so we need to disrupt the cell. This is done using alkaline lysis, which solubilizes the membrane using an alkaline solution of sodium hydroxide and STS, which releases our plasmid DNA along with other intracellular material. So at this point, we need to get rid of a mass of solids. We use dead infiltration to do this, which allows 67% plasmid recovery while removing 99% of the solid mass. Now we need to further purify our product, since the plasmid DNA is very dilute at this point, with over 80% of the solute mass being proteins and RNA and other DNA that we don't want. To do this, we use isopropanol precipitation. This is just done in a standard bioreactor. So we precipitate all our DNA and we centrifuge it down again. And then we follow with a subsequent second precipitation step. So this, once again, precipitates the DNA while removing proteins to almost undetectable levels and removing endotoxins 20,000 times more than just one precipitation step. This increases our plasmid purity 6.5-fold. So once again, we centrifuge that down and we proceed with our final purification, which, since we're creating a pharmaceutical product, we need a very high purity. To do this, we first use hydrophobic interaction chromatography. So this uses mildly hydrophobic ligands to separate molecules on the basis of hydrophobicity. So this allows us to separate our plasmid DNA from things such as genomic DNA, RNA, and endotoxins. We follow that with the size exclusion chromatography, which allows separation of supercoiled plasmid DNA and open circle plasmid DNA, because we want supercoiled plasmid DNA so that when we deliver this as a therapy, we can deliver more of it in less of an injection or less of a treatment. So this enables the recovery of almost pure supercoiled plasmid DNA, and then we finish with just a standard filtration sterilization step. Like I said, we use SuperCode Designer, so we obtain these equipment costs from SuperCode Designer, which are based on the average market values for the various equipment that you see here. And we came up with a total purchase cost of $2.5 million. So this was used to calculate how much it would cost to start this facility and the direct fixed capital that we would need. So this is done by using multipliers to calculate direct costs such as installation of equipment, process piping, equipment instrumentation, along with indirect costs such as the engineering and construction. And then we added contractors' fees and contingency finances that you earn. So like I said, you start with your purchase cost of your equipment, which is $2.5 million. Multipliers up with a total of $8.8 million, which was then used to calculate the indirect costs of the engineering and construction. This was a total of $5 million, which was added to our contractors' fees and our contingency finances. And we would require a direct fixed capital cost of $16.3 million to start this entire facility.
So at the end of our project, we had hoped to design 3 engineered constructs of baleen, cysteine, and thiamine, and compare that to the actual wild-type activity of the naturally occurring construct, as well as a design scale-up and a finished final design report and poster. So we would like to give a special thanks to all those at the Milwaukee School of Engineering, including our advisors, Dr. Hesmer and Dr. Afshan, as well as those at the Milwaukee Health Department and Medical College for their providing of kids' materials and facilities. And at this point in time, we would like to thank you for your time and attention and open the floor to you guys for any questions you might have.